Just like for glycolysis, we will be going over the process of acetyl-CoA formation and the citric acid cycle. Then I will go over what you should remember for these metabolic pathways. Acetyl-CoA formation and the citric acid cycle take place inside of the mitochondria of eukaryotic cells. If we are talking about prokaryotes, it happens in the cell membrane. ATP is generated by substrate level phosphorylation or by moving phosphate groups from molecule to molecule. Energetic electrons with their protons in the form of hydrogens are carried by NADH and FADH2. When the pyruvate from glycolysis is moved from the cytosol to the mitochondria, it is converted to acetyl-CoA. In the process, we clip off an acid group. The hydrogen from the OH end of the molecule is removed and placed on NAD+. The rest of the acid group leaves as CO2. The acetyl-CoA then goes into the citric acid cycle. In the process, two more acid groups are lost as CO2, which generates two more NADHs. Another NADH is generated later on in the cycle. FAD is related to NAD+, but it can carry two hydrogens, whereas NAD+ can carry only one. And ATP is generated. In the process, the cycle returns to its starting point molecule, which can then pick up another acetyl-CoA and go around the cycle again. So here's what I want you to remember for acetyl-CoA and the citric acid cycle. Why does the cell perform acetyl-CoA formation and the citric acid cycle? What is the benefit to the cell? Where does it happen within the cell? What is the starting substrate or substrates for the metabolic pathway called acetyl-CoA formation and the citric acid cycle? What energy is used during the metabolic pathway called acetyl-CoA formation and the citric acid cycle? What energy is produced? What are the ending products for acetyl-CoA formation and the citric acid cycle? Once again, search for the answers, then ask me about it in lecture.